All right. Um, Good morning, everyone. Welcome to the workshop on COVID-19, a novel coronavirus genomics workshop. In this workshop, we'll speak about genomics data analysis and try to understand how analysis tools can help detect viral genomes and next-generation sequencing data. Here, we're going to learn how to distinguish between viral strains, identify specific functionally significant changes in viral genomes, and we'll be discussing virology from a data perspective. Let's start with the name itself, COVID-19. This name stands for coronavirus disease, and 19 is for the year that this epidemic started, which was 2019. In this workshop, we will try to touch on these factors: the coronavirus itself, the disease, and how it is a result of viral transmission from animal reservoirs, as well as the entry into the human cell. We will be discussing the collected data that we have from 2019 and the first 3 months of 2020. We will speak about all of these from the perspective of bioinformatics, which allows us to study biology, or in this case, virology as a data science. Together with our collaborators, we've been building a growing online community of bioinformaticians in training, ranging from high school students to faculty and researchers at top universities around the world. Our team offers big data analysis tools and a quality learning resource, such as courses, projects, and tutorials. We have programs that are designed around various topics, which discuss bioinformatics approaches for processing, analysis, integration, and the interpretation of multi-omics data from oncology, neuroscience, agrobiology, as well as infectious diseases. I encourage you all to sign up for our free courses, as well as the ones that are on our online portals, and start learning or teaching bioinformatics. It is one of the best ways that we can explore the world around us at the molecular level, and there's going to be zero equipment or lab costs associated. Last Friday, we met to review the basic concepts that we needed to find, understand, and start thinking about publicly available data sets, as well as the tools for their analysis. And I hope that you enjoyed that session. If anyone wants a recording of that first webinar, please let us know. This week, we're going to be holding 2 additional sessions that we're going to be making available to the public. In these sessions, we'll continue to speak about sequence analysis, and we'll go deeper into the world of coronaviruses. Looks like we still have people still trying to join with their video and sound. I'll do my best to kind of try to moderate that. I'll have to admit I have attention deficit disorder, so I may have to remove you from the room if you distract me too much. So anyways, today we're going to be continuing to speak about variations in sequences that we can find by analyzing data. In our next session, which is going to be on Friday, March 27th, we'll start exploring the way that sequence changes are affecting protein structure and function. Sequences of full genomes, individual reads from samples, or specific amplified regions can be used to study natural variation, diversity, changes over time, and find changes in RNA or DNA that can be linked to various diseases. Can also be looked for things that are involved in other types of traits or phenotypic attributes. Such links can be direct, such as the change in structure as a result of the non-synonymous mutation, and these are changes that occur at the protein surface or other type of chemical property. Or it can be indirect, for example, silent changes or insertions that change the way a process that has many steps is modified slightly to produce more significant changes. Today, we will speak about these kinds of changes and methods of analysis that we can use to find changes in sequences, and then annotate them, as well as link them with phenotypic data. In today's session, we'll speak about topics from our full program that's dedicated to bioinformatics of infectious diseases, where we'll analyze data from public domain sources and learn to interpret the analysis results to learn about various aspects of viral infections. The full the full program will start on May 4th, 2020. Be sure to check it out by going to the link that I'll be posting in the chat. In today's session, we're going to be speaking about the novel SARS coronavirus and explore its origin as a zoonotic disease. A zoonotic disease is a disease that comes from animals to humans in a process called spillover. We will learn about the process of endocytosis, by which the virus enters the human cell to replicate, and then hijack the cellular machinery for its protein production and replication. We'll also be learning about several data analysis approaches for genomic data, including detection of viral genomes and NGS data, the identification of viral strains and haplotypes, finding mutations that characterize viral strains, and then interpreting its significance. Finally, in order to apply various methods of analysis, we'll need a lot of data. So we'll be exploring data sets that are related to current and previous strains of coronaviruses, such as SARS, MERS, and the alpha and beta coronaviruses. Coronas in humans, animals, and in various geographical locations tend to be different. And so we're going to be talking about host metagenomic samples, as well as host response data that can help us to detect presence of viral strains, discover new strains, or to understand the function of various viral proteins. This brief session will only be an introduction because the range of topics is broad and comprehensive. If you understand bioinformatics and you're interested in this area of bioinformatics, please make sure to sign up for the full program, where the same topics will be explored in depth and then applied to the study of several recent viral pandemics, such as Ebola, flu, and enterovirus D68. Additionally, we'll talk about infections such as tuberculosis that are of bacterial origin. To start speaking about these topics, let's first review the basic terminology that help us to study virus from a bioinformatics perspective. This is also a data perspective. Bioinformaticians help extract biologically meaningful information from data that's often complex. And it requires many analysis steps to explore patterns, develop questions that can be asked from our data, and then ask questions using analytical methods. And then the final step of that is to find answers that can be validated. So in this workshop, we're primarily going to be speaking about genomic data and how it can help us to study a virus, which is namely the pathogen behind COVID-19. This is a disease that's caused by a single-stranded RNA virus that's called severe acute respiratory syndrome coronavirus 2 because it belongs to the coronavirus family. It's called a corona because it has a crown-like appearance that's formed by its spike surface proteins. Other coronaviruses are capable of causing illness, ranging from the common cold to more severe diseases such as MERS. This is also the 7th known coronavirus to infect people, after a couple of strains that I'm going to read out for you, which would be 229E, NL63, OC43, HKU1, MERS-CoV, and then the original SARS-CoV. Analyzing the genomic data from publicly accessible repositories allows us to learn about the origin, structure, and function, and how this pathogen is different from its family members. The common human coronavirus usually causes a mild to moderate upper respiratory tract illness, such as the common cold. Most people get infected with one or more of these viruses at some point in their lives. So there's a couple of human coronaviruses that we have to talk about. Remember earlier I said that there was the 229E, which is alpha coronavirus. There's also several beta coronaviruses, such as OC43 and then HKU1. There's also MERS, SARS-CoV, SARS-CoV-2, and then that's the novel coronavirus that causes coronavirus disease, and it's also called COVID-19. People around the world commonly get infected with human coronaviruses such as alpha and beta coronaviruses, and so these are the most common types that we'll be talking about. So just as a recitation, the alpha and beta coronaviruses are going to be 229E, NL63, OC43, and HKU1. Sometimes coronaviruses infect animals, and they can evolve and make people sick, as well as become a new human coronavirus. So 3 recent examples of this are going to be the 219 nCoV, SARS-CoV, and then MERS-CoV. So looking back through this, these are the different types of coronaviruses that we have. So as we can see, there are Delta, Gamma, Beta, Alphas, and then our human SARS-CoV-2 is a beta coronavirus. Before 2002, it was thought that coronaviruses cause only self-limiting and mild infections of the respiratory tract in human beings. 2 of the coronaviruses that are considered during this time were beta coronaviruses, and the other 2 were alpha. These coronaviruses were endemic, which means they're spread in certain geographic areas and cause about 15 to 30% of the infections of the respiratory tract each year in the areas that they're endemic to. The SARS outbreak that happened in China during 2002 to 2003 was caused by SARS coronavirus, which was a beta coronavirus. At the time, this outbreak was amongst the most severe illnesses that had occurred due to a coronavirus, and more than 8,000 people had the illness. During this epidemic, researchers obtained viruses that were closely related to the SARS coronavirus from various exotic animals, such as raccoon dogs and the Himalayan palm cavets. But it's generally accepted that SARS coronavirus was first found in bats. This virus is zoonotic because it spread between humans and animals. And the SARS coronavirus outbreak is controlled in the year 2003. And since 2004, there haven't been any known SARS cases reported anywhere in the world. After that, a new human coronavirus appeared in 2012 in the Middle East, and this was called MERS, which is another type of beta coronavirus. It's caused in succession of highly pathogenic infections of the respiratory tract, and this is endemic in the Middle East. During the initial stages of the outbreak, the illness was severe in many cases, but the outbreak didn't accelerate until 2013, with only a few cases occurring before that. Detailed investigations found that MERS coronavirus was passed from camels to human beings, and that SARS coronavirus passed from civet cats to human beings. Both camels and cats were acting as intermediate hosts and transmitted the coronavirus to humans and bats. It's important to ask questions that can be answered with data. Typically, this data has to be analyzed and separated from signal and noise, and then we'll be able to find patterns that are relevant to the question that's being asked. Some of this data is simple. For example, metadata such as who collected the sample, or which type of location it was taken in, or the date. There's not a lot of detail in such data, and it's prone to misinterpretation as well as inaccuracy. However, genomic data is very detailed and accurate most of the time. It can offer deep insights into precise answers if the questions are asked correctly. Once we do that, we can discuss how these patterns and data can be interpreted. And this process is called extracting meaning from data. And it's the core of bioinformatics. And as we go through the session, start thinking about a question that you want to ask. To start, let's take one of these questions, such as where did this novel coronavirus come from? Or maybe which animal, location, or how did it start infecting humans? And how do we know? To answer this question, we have to find data patterns in the available genomes of coronaviruses that can help us to answer this question. NCBI has a submission of this novel coronavirus genome with metadata included. Here it was submitted as January 5th, 2020 from China, and the collection date was December 2019. I'll post a link in the chat to where you can find this data on your own whenever you get a chance. So here we can go onto the website and find some novel data about our sequence. To do so, we can navigate NCBI virus database and browse the publicly available data. You can simply search NCBI virus or go to the link that I'll post. And let me see if I can get that up for you. So as you get here, one question you might be asking is which virus tax ID should we pick? And the answer to that is you would like to pick severe acute respiratory syndrome related coronavirus, which has a tax ID of 694009. So it's going to be the penultimate one right here in the bottom. We can pause now and see if anyone has any questions about getting to the website. And then I'm going to try to push through some of these slides and save some questions for the end, so that way we can get through the material. So just to reset these steps, we're going to go to this link, and then we're going to look for severe acute respiratory syndrome related coronavirus, which has a tax ID of 694009. And so we've gotten this far here, and then we can see that there are going to be several strains. So as you can see, you find over 1,500 sequences that are available for us to study, compare, and interrogate. So the question we must ask about the specific sequences that we would like to select for our comparison, or when we do so, we have to revisit an important topic that we started speaking about last time. What is the question that we're trying to ask? Recall that last time we started following an example found in research publications where 6 sequences were compared. There were 3 from bats, 1 from pangolin, and 2 from human hosts. Let's try to find a similar sequence in this database. We can select all of the 139 sequences by selecting the checkbox at the top of the list. We have one question, which would be our reference genome? So what would the reference genome be if you were selecting for SARS? Would it possibly be humans? As I was saying, then we can build a phylogenetic tree that shows relationships between these sequences in a cladogram, which is a tree-like dendrogram. A phylogenetic tree shows relationships between present-day species based on both common ancestors and time. It has nodes, branches, clades that depict the evolutionary distance between taxa that we have sequences for. This tree can be horizontal, radial, or circular. And as we see in our tree, we have too many branches and nodes to understand the relationships between these sequences. So what we would need to do is go back and then reduce the number of genomes and then run this analysis again. This time, let's select a few samples from a few years ago and then compare them with current samples from bats and humans. So bats are mammals of the order Chiroptera. And then there's a family of bats commonly known as horseshoe bats. In addition to single living genus, the Rhinolopus is one extinct genus. There's also Paraliencyteris, which has also been recognized. Horseshoe bats are closely related to another type of bat called Hippociderae, which are sometimes been included in with Rhinolopteridae. We can add a few samples from 2005 to 2006, and a few from 2018, and several from 2020. As a result, we have 7 different genome sequences from USA and China, from bats and humans. And let's see what kind of tree we can get now. Your tree should look something like this. And as you notice, the information this tree gives us is limited because we add year and host data from the previous table. Please take a couple of minutes to review the accession numbers and find what year and species the samples are from. So what do you think this tree shows? What type of information, species, location, and year can help us interpret this phylogenetic tree? Mohit says China and USA. So let me add this information on the slide so that we can quickly see the answer. So here we have the year of collection. So good job, Emersabe. And yes, we do see that USA is clustered together here. Now let's align these sequences and see how alignment works. So you can click on the align box there and it will align your sequences. Multiple sequence alignment, or MSA for short, is generally the alignment of 3 or more biological sequences, which can be protein or nucleic acid of similar length. From the output, homology can be inferred, and the evolutionary relationships between the sequences can be studied. By contrast, there's pairwise sequence alignment tools, which are used to identify regions of similarity that may indicate functional, structural, structural or evolutionary relationships between two biological sequences. Some of the commonly used tools for MSA are called Clustal-Omega, K-line, MAFFT, Muscle, and these are going to be described as follows. Clustal-Omega is an MSA tool that uses seeded guide trees and HMM profile techniques to generate alignments. This is suitable for medium or large alignments. K-line is a very fast MSA tool that concentrates on local regions. It's suitable for larger alignments. MAFT is a tool that uses fast Fourier transforms, which is suitable for medium large alignments. And then Muscle is accurate, and it's especially good with proteins. This one's also suitable for multiple or medium alignments. We have a couple of other entries from our participants. We have Prank and Eugene, and there are many different types of alignment tools. So here you would want to select tools and then coloring. There's a frequency-based difference. How many times a change in sequence is found? The more changes, the more red that we'll see. The key is to know what they're good for. Some for short and some are for long sequences. Also on here, you need to select Show Differences. And this method highlights differences from the master sequence. Let's take a look at the result we want to see that might be more significant of these changes between our genomes. If we expand the genomes that we have an annotation for, we'll start seeing that the genome is not just a string of letters. These regions code for proteins that the virus will produce. You will see the gene name in some cases and name of the encoding protein. So some more questions that we can ask are, which gene has a lot of differences between genomic sequences? So I think the answer depends on what we call difference. Are we talking about substitutions, deletions, or insertions? And how do we quantify such differences? Another question we might ask is, what are other major differences that you observe in these different genomes? This might be that some are more annotated than others, and some have visible gaps in their genomes compared to the others. Let's talk a little bit about the coronavirus genome itself. So what does the coronavirus genome encode? We can explore the sequence at the nucleotide level to understand how the virus genome is organized, and then we can find out the sequence variations we observe. So speaking a little bit more about the genome here, we have coronaviruses that are enveloped positive sense RNA viruses, and they're characterized by club-like spike proteins that are projected from their surface. They have an unusually large RNA genome, as well as a unique replication strategy. These contain a non-segmented positive sense RNA genome that's around 30 kilobases long. And this genome contains a 5' cap structure along with a 3' poly A tail. This allows it to act as its mRNA for translation of the replicase polyproteins. The replicase gene encoding the non-structural proteins occupies 2/3 of the genome, which is about 20 kilobases, as opposed to the structural and accessory proteins, which make up only about 10 kilobases of the viral genome. At the 5' end of the genome, there's a leader sequence and an untranslated region that's called a UTR, and this contains multiple stem loop structures that are required for RNA replication and transcription. Additionally, at the beginning of each structural or accessory gene, there are transcriptional regulatory sequences that are required for expression of genes. This 3' untranslated region also contains RNA structures that are required for replication and the synthesis of viral RNA. The organization of these coronavirus genomes features a 5' leader untranslated replicase, S, which is the spike protein, E, which is for the envelope, M for the membrane, N stands for nucleocapsid, and then remember that there's the 3' untranslated polyadenylated tail. There are also accessory genes that are interspread within the structural genes at the 3' end of the genome. These accessory proteins are almost exclusively non-essential for replication and tissue culture. However, some have been shown to have important roles in viral pathogenesis. So here we can see that the sequence of the proteins of the novel coronavirus under the accession number NC045512. I'm going to post that in chat for you to look at. Virus nomenclature is a formal system of names that's used to label viruses and taxa. The fact that there are names for nearly all viruses within the species is due to the historical perception that viruses as causative agents of specific diseases in specific hosts, and to the way that we visualize and catalog and then classify newly discovered viruses, which increasingly includes viruses that have not been linked to any known disease or in their respective hosts. The World Health Organization, which is an agency of the United Nations, coordinates international public health activities that are aimed at combating, containing, and mitigating the consequences of communicable diseases, which include major virus epidemics, and it's responsible for naming diseases that are caused by newly emerging human viruses. In doing so, the World Health Organization often takes a traditional approach of linking names of specific diseases to viruses and assessing virus novelty by an apparent failure to detect a causative agent using established diagnostic assays. Apart from disease, geography, and the organism from which a given virus was isolated, these also dominate the nomenclature, occasionally engraving connections that may be accidental rather than typical, or even stigmatizing, which can be avoided. So here we can see a virus species names are often very similar to the name of the founding member of a respective species, and they're frequently confused in the literature with names of individual viruses of their species. You'll also notice that the species name is italicized, and it starts with a capital letter and should not be spelled with an abbreviated form. Hence the species name severe acute respiratory syndrome related coronavirus. In contrast, this convention does not apply to virus names. So for instance, there's severe acute respiratory syndrome coronavirus, or SARS-CoV, as it's widely known. So essentially what this is saying is that based on the type of virus, there's going to be different types of naming schemes. And one of the main goals is to not stigmatize certain groups. Based on the number of available sequences, we can start asking a lot of different questions because we already know about the pathogenesis, origin, or replication of a previous virus. So for instance, SARS or MERS from the previous decade. Based on comparative analysis of such sequences, we can derive how the current genome is different, as well as what implications that might have on various properties of its proteins. A challenge in analyzing so many genomes at once and comparing them at multiple levels occurs because we're doing this from nucleotide, amino acid, and then organizational properties. And this challenge is that the simple tools on NCBI do not allow for such flexibility or processing power. So in our upcoming sessions, we're going to be leveraging server.t-bio.info platform And we're going to be running big data sets and then comparing how the various sequences differ at the genomic level. And we're going to be studying mutational fitness as well as virus phylogeny. So I see that Mohit has posted the link in the server, and I encourage you to take some time between now and our next session to go on the server and utilize that resource. One such analysis that we can perform is an advanced Bayesian phylogenetic analysis, such as BEAST. It uses the birth-death process, which is most popular in evolutionary studies. Another is called Yule, and it's a birth-death special case. And here it's expected that organisms are dying and the population size is not constant. In the virus case, the size is almost constant, and therefore coalescent constant size might be a good option. But by running such an analysis, we'll find that the COVID-19 genome is closely associated with SARS. Consistent with previous reports, SARS-CoV-2 clusters with SARS-CoVs in trees of the species SARS-related coronavirus and the genus beta coronavirus. The distance estimates between SARS-CoV-2 and the most closely related coronaviruses are going to vary amongst different studies depending on the choice of measure. And this could be whether nucleotide or amino acid or the genome region. But genomes don't only vary between their species or well-defined strain. There's also a lot of variation that occurs during the pandemic. Viral genomes collected from various locations, dates, hosts during a short time span can reveal this trend. If you would like to see a pandemic in action, you can visit a website that I'll post in the chat. It's called nextstrain.org. And I'm actually going to hop out and show you this website because I think it's really cool. So in here, you can kind of go in and zoom and see what's going on, for instance, in the United Kingdom. Or for instance, what's going on in Southeast Asia. We can also look at phylogeny and click on different information to see what's going on with that. We can also switch the date range and see that how that affects our diagram as well as our map. And so anyways, between now and next session, I encourage you to go on this website as well and to play around with the various parameters here and kind of see what type of maps you can generate. Very cool. To find mutations within a pandemic, such as SARS-CoV-2, we can select for genomes specifically during this time period and then analyze them for variability at different levels. But a global sequence alignment is not the full story. For example, in recent reviews, even a small number of genomes can reveal specific changes that occur between genomes that we need to be looking for. So here you can see the relationship between recent and more distant human and bat genomes with SARS and novel coronavirus SARS-CoV-2. The challenge that we'll turn to next is the identification of specific ways these genomes are different by looking at the spike glycoprotein structural subunits that have specific patterns that are important for its function, as well as the adaptation between animal and human hosts. For us to start answering this question, we have to understand the data and how to find it and find meaningful biologically interpretable data, as well as the details in the sequences. So let's start by clarifying some of the terminology that we'll use in analysis. FASTA and FASTQ are different types of files that you'll be importing into the various pipelines that we'll be running. There's also nucleotides, which are obviously the types of bases that are involved in genomics. There's substitutions, insertions, and deletions, which are going to be various types of polymorphisms that can occur with nucleotides. There's amino acids, which are going to be the product, that are going to be linked as the product of translation. Obviously, there's mutations, which are variations that occur. You're also going to be reading about zoonotic transmission and spillover, which is what happens between animals and humans. And then we have other terms such as cell entry, phylogenetic relationships, and then obviously DNA, RNA, and protein. So these are some terms that you can look at as you're reading, especially if you're at a beginner level and wanting to gain some more information. Genomes are assembled from next generation sequencing, which is typically done using a process called shotgun sequencing. This process includes shattering the source material, which is going to be RNA or DNA, and then it's going to be sequencing of short reads that are 30 to 300 base pairs long. These sequences are going to be assembled into a full genome by looking at the consensus sequence. In other words, if the sequence of reads all shows the exact same letters, they're going to be considered as a consensus. If there's a disagreement between these, the majority is going to win. And as a result, from a single isolate, we can get the full sequence of a viral genome that's going to be most abundant. This process is fairly accurate. However, it reduces the diversity of potential present variations of the virus in each sample to a single sequence. The more samples that are sequenced, the higher our confidence that the strain has a consistent characteristic. After these sequences are found, we can start looking at the genomic characteristics, such as our genes, or special coding sequences that can be associated with a function. Let's first speak about the sequence itself. We can talk about point mutations, and as well as the genetic code and how amino acids are linked together to form proteins. When we compare sequences of two genomes in a FASTA file, we will be looking for variation. Typically, we'll be looking for a single point mutation or what's called a single nucleotide variation. These can be substitutions, insertions, or deletions. Such changes have differential biological significance because a single nucleotide change can result in several changes in the codon, which is a set of 3 nucleotides strung together that are recorded in mRNA, which will be translated into protein. Remember that proteins are our functional element. A gene that's typically recorded in DNA form is going to be converted through transcription into mRNA and then translated into protein. This machinery is what's folded into the functional product. And here it's absolutely true that structure is indicated of a function. We're briefly going to go over the various classes of building block amino acids that comprise a functional peptide. So there's 20 standard amino acids that occur in nature. They can all be divided up mostly between 3 classes. These are going to be polar charged, polar uncharged, and non-polar. Let's start with the non-polar amino acids. The non-polar amino acids are generally going to be found in the interior of peptides. And these are generally going to be structural types of amino acids. The polar uncharged are typically going to be at the cell or the protein surface. And these are polar, but they do not possess a charge. Generally in aqueous solution, they don't have any type of acid-base properties. The polar charged, however, do possess either a negative charge or a positive charge. And these types of amino acids generally occur in the active site of enzymes or at some functional aspect of the protein. So generally, mutations that will change, for instance, a glycine to a valine won't have a deleterious effect. However, if you were to change an aspartic acid to, say, for instance, a valine, that would cause a very deleterious effect, because this is usually going to be found bound to its counter ion, which would be a positively charged amino acid. So we can talk a little bit more about cell structure as well as where these amino acids might be found. For example, the chemical properties of amino acids explain how membrane proteins function. So here I'll talk about lipid bilayers as well as integral membrane proteins. So here we can see the outside of the cell, which is also called the extracellular space. And here we have different types of insoluble molecules, and then carrier proteins that are involved in transporting these either with or against their concentration gradients. On the inside of the cell, this is called a cytoplasm, or if it's referred to in literature in reference to metabolism, called the cytosol. And these different types of molecules are brought in for some biological purpose. If the protein is one that spans the membrane, which is generally around 70 angstroms long, it's called an integral membrane protein. And there's also types of proteins that are called carrier proteins that are involved in passive diffusion or facilitated diffusion. The bilayer has two different parts to it, one of which is going to be a phospholipid head. These phospholipid heads are polar in nature. And so as we go through our amino acids, you'll see that generally a polar or charged amino acid is going to be at the surface of this protein right here. Because the bilayer is also nonpolar in nature on the inside here, the types of amino acids that would be present in this protein in these regions would be nonpolar in nature. So again, these are the polar charged amino acids. These are hydrophilic, meaning that they want to bind to water, and they also possess a charge. These are the amino acids that you'll find that are going to function in binding and catalysis. And so whenever you read about the COVID or SARS disease and the virus, you'll find that there are different proteases that are involved with it. Oftentimes that the mechanism of that action is going to be produced by way of the different types of amino acids. And so as you go through, you'll find that a lot of these residues are going to be very important for that. Another type of amino acid that we talked about was uncharged polar amino acids. These are going to be threonine, serine, cysteine, glutamine, tyrosine, and asparagine. Remember that these amino acids are typically found on the outside of the peptide. And some of these also function in binding and catalysis. One notable one would be the serine proteases. And then the ones that are found on the interior of peptides, where they minimize their contacts with water, are going to be hydrophobic. These types of amino acids are generally structural. And for instance, some of them produce kinks in the protein structure. For instance, proline is what's known as a helix breaker. Other types of amino acids function in other structural things such as disulfide bonds, which would be between cysteine residues. And as you go through, there's a lot to know about amino acids, but I think the general take-home message from this should be that they can be broken up into 3 different classes, and where they reside in a protein is going to be dependent upon their structure and their proclivity to bind to water or not. Proteins also have to be folded, And this is going to be done in a manner that follows thermodynamics. So the way that proteins fold is going to be governed by the fact that it's in an aqueous solution. And so with similar proteins from different organisms, the structure tends to be more conserved than the exact sequence of amino acids. Mutations that occur are natural variations and tend to be on the exterior of proteins. Many of our functional changes can be studied from analysis of structure, as well as structure similarity. Proteins are folded post-translationally and create the 3D structure of a peptide. Another concept you'll be reading about are motifs, which are simple combinations of secondary structure elements, which are also referred to as super-secondary structural elements. And so here we can see a beta-pleated sheet that occurs, or an alpha helices. These are both called secondary structure. The actual sequence of amino acids as they occur in a peptide is what's referred to as the primary structure. And then how these fold after translation defines the tertiary structure. One other term that you might see is quaternary structure, and this is where subunits of various peptides are going to be interacting with each other. One example I can talk to you about that has quaternary structure would be hemoglobin, which has 4 different subunits linked to it. Myoglobin is an example of a protein that does not have a quaternary structure because it is only one of these subunits. So when we study these variations between viral genomes, we have to remember that the changes that we observe at the sequence level will have different impacts on protein structure and function. If we want to understand a virus and how different in terms of infectivity, being the entry into a new host, or how effective it is at replication, we have to consider the protein product. There are several different ways that we can study protein function. This can be based on the amino acid sequence, the 3D crystal structure, or high throughput data that's available from this protein interacting with a host cell. So here we can see a peptide going from linear to a folded form. And then know that we can study proteins based on amino acid sequence, the 3D structure, or the data that we get from gene expression. Let's look at several project data sets where we can see some of these methods that are applied to real data sets that are produced from these outbreaks. Let's talk about where it all began. In the full program, we're going to be looking at a data set where the first novel coronavirus genome was detected and assembled. The project is titled The Detection of Viral Genomes from Human and Bat Metagenomic Sequencing. Here, the identification and characterization of the new coronavirus occurs, which has caused an epidemic of acute respiratory syndrome in humans from Wuhan, China. This epidemic started on December 12th, 2019, and at the time, it caused 2,794 laboratory-confirmed infections, including 80 deaths by the 26th of January. Full genome sequences were obtained from 5 patients at the early stages of the outbreak. The sequences were almost identical and shared 79.6 sequence identity to SARS-CoV. Furthermore, it was shown that 2019 nCoV is 96% identical to the whole genome level, that of the bat coronavirus. Pairwise protein sequence analysis of 7 conserved non-structural protein domains show that the virus belongs to the species of SARS-CoV. In addition, 2010 nCoV virus was isolated from the bronchoalveolar lavage fluid of a critically ill patient that could be neutralized by sera from several different patients. Notably, the authors confirmed that 2019 nCoV uses the same cell entry receptor, which is called ACE2, and that's short for angiotensin converting enzyme 2. Let me post a link to the paper here for you to take a look at between now and next session, where we're going to talk about proteins. So here we have the spike glycoprotein, and its genome has been sequenced, and that genomic information was promptly released. Despite a high similarity with the genome sequence of SARS-CoV and the SARS-like CoVs, we identified a peculiar furin-like cleavage site in the spike protein of the 2019 nCoV, which lacks in the other SARS-like COVIDs. So there are possible functional consequences of this cleavage site in the viral cycle, as well as the pathogenicity and its potential implication in the development of antivirals. Another project is the viral protein function. The title for that is the release of severe acute respiratory syndrome coronavirus, nuclear import, which blocks and enhances host transcription in human lung cells. The purpose of the experiment was to compare transcriptomics of 2B4 cells, which are clonal derivatives of Calu3 cells that were infected with either IC SARS-CoV or the IC SARS Delta or 6 mutant of that virus. They had a pretty unique experimental design. The Calu3 cells were infected with either IC SARS-CoV or the IC SARS Delta or 6 mutant. The sample cells were collected at various intervals in the post-infection phase, and then each infected sample was done in triplicate. And so triplicates are defined as 3 different wells, which are plated at the same time, using the same cell stock for all replicates. There are triplicate time-matched mocks for each time point from the cell stock as the rest of the samples. And then the culture medium, which is the same as what the virus stock is in, was used for the mock infections. I'll also post that link in the chat for you to peruse at your convenience. So on Friday, we're going to be discussing protein changes as well as viral entry. And we're going to talk a little bit more about coronavirus biology, specifically the entrance of the virus into the cell and how this virus replicates. We're also going to be talking about the analysis of viral proteins, and then mapping nucleotide sequences on protein structures that interact with human proteins. So I look forward to seeing you on Friday, and we can have that discussion then. I have one more thing to talk to you about today, and it is our Gene Lab, which is an option that you have for purchasing a research license on the T-bioinfo research platform. It includes 5 terabytes of storage and 3 hours of support with one of our specialists. I'll also post that link in the chat for you to take a look at. And I have to mention that in the month of April, we're going to be offering a 50% discount for research licenses and educational licenses due to the coronavirus outbreak. So please take advantage of that. And I look forward to seeing you on Friday. I hope that you all have a wonderful week.